Our first speaker, Manuel Montano, is a new assistant professor in environmental science at Western Washington University. He's doing really cutting-edge work on identifying microplastics in the environment. His research group focuses on the development and application of new analytical tools and methodology to study the nano-environment. His current research examines a broad array of topics, from microplastics to engineered nanomaterials, as well as fundamental studies into nanomineral behavior in aquatic systems.
So thank you for the introduction. As mentioned, my name is Manuel Montano. I'm a professor up at Western Washington. And today what I'm going to talk about is some of the approaches I'm taking to study the behavior of microplastics in the environment. And with that, really what I'm doing is I'm bringing in my experience with quantifying and characterizing nanomaterials in the environment. And really both of these issues are what I consider these sort of needle in a haystack problems. And when it came to nanomaterials, one of our first questions was just first determining, are there nanomaterials in the environment? So can we find those needles present in the haystack? And then once we moved on from that, we started to ask ourselves, how big are those needles, right? As the size really determines the toxicity and the behavior of nanomaterials, and how many are there? The particle number concentrations are almost more important than the mass concentrations of nanomaterials. And then as we get further along, now we're starting to ask ourselves, okay, well, how do the needles and the straws of hay, how do engineered nanomaterials and naturally occurring nanoparticles and colloids interact with each other? And these are the same questions that we're trying to address with microplastics. Are there microplastics out there? How many are there? How big are they? And then further down the line, we hope to ask ourselves, how are they interacting with other naturally occurring colloids and biomolecules in the environment? And if we consider the complexity of this issue, again, I'm going to use nanomaterials as an example. When it came to engineered nanomaterials, we had an environment, and we have these three broad classes of nanomaterials. We have our engineered nanomaterials, the ones that we create specifically to have nano properties, and so those can enter into the environment through use and disposal and agricultural practices. And then we have incidental nanomaterials, which are also man-made but not intentionally so. So this is the colloids that come from stormwater runoff and waste disposal, urban dust, brake wear, tire wear. And then all of those particles are swamped in comparison with the amount of naturally occurring nanoparticles and nanominerals that are present in the environment, which are formed from the erosion of rocky material, neoformation, sea spray, and atmospheric deposition. So even though we're talking about microplastics today, I'm just trying to draw your attention to the fact that this is a colloid science problem that I'm really trying to address. How can we specifically identify microplastics in the environment? So we need techniques that are both sensitive to the low concentrations that we're expecting, as well as selective to the specific kind of particles that we're trying to identify. And when it comes to identification, we're also still developing our methods there. So conventional conventional analysis usually would involve filtering of an aquatic sample, and that filter cutoff would determine what is particulate and then what is dissolved. But we know that the operational 450 nanometer filter cutoff, there's a lot of particles that are smaller than 450 nanometers. So to say that they're dissolved is kind of a misnomer. And so with that, we've developed new techniques in the time of nanomaterials that help us identify individual particle characteristics on a single particle basis. Techniques such as field flow fractionation and single particle ICPMS, as well as using things like electron microscopy, here identified as EM. And then beyond that, again, trying to address the interactions of naturally occurring and engineered nanomaterials. We now have emerging techniques such as single particle ICP time of flight mass spec, as well as liquid transmission electron microscopy. And so again, what I'm trying to point out here is that we're moving from trying to identify the whole haystack and trying to tease out concentrations to looking at the individual particles to try and ascertain some information. And then finally looking at the whole collective as a whole and how they interact with each other. Now, when it comes to microplastics, we're a little, I wouldn't say behind the curve, but we're struggling to catch up to the levels of accuracy and precision that we need. So the common techniques that we use for microplastic identification, conventional light microscopy is the most common, where we just visualize microplastics. But as you can imagine, this has a very limited size resolution, despite being low cost. Scanning electron microscopy is going to offer some higher magnification as well as better resolution, but it's going to be very low throughput and require a lot of preparation in order to get good images. The other consequence of this is that both these techniques won't be able to give you information about how the microplastic is behaving in situ in the environment, right? They need to be dried, they need to be filtered, and you lose a lot of information about how they interact as particles when you try to image the particles as a consequence. We also have spectroscopy techniques such as FTIR and Raman spectroscopy. Both these techniques are important because they give you chemical information about the microplastics. It's not enough just to know that there's a plastic particle. It's very important to determine the composition and the properties of the particle, as that's going to determine its fate and toxicity. And so we need to find a way to utilize these techniques, but the problem is that we're limited in terms of the sizes that we can detect down to. So here I have an image that's showing we have our bulk plastic product, which can break down into smaller particles that we can visually identify or use light microscopy to identify. Once it gets smaller and smaller, we can use things like FTIR and Raman, or couple microscopy to FTIR and Raman for micro FTIR and micro Raman. But again, we're limited in terms of how small we can go down. So we're only really able to get down to about a micron in terms of the particle sizes that we can detect and characterize. And so there's this whole region as we're approaching the nanoscale that we can't even begin to try to identify. And so that's really an area that we're trying to develop and bolster right now in order to better understand how nanoplastics behave in the environment. And so that is some of the background to get me into my work. And my work really started because I came across a paper from a colleague of mine at EABOG in Switzerland, who was using metal-doped nanoplastics to investigate how microplastics move through the environment. And so what Denise Matrano, who is now at ETH Zurich, did is she labeled these polymer particles with palladium metal. And so it's kind of hard to see here, but you can see these little red dots inside the plastic correspond to palladium that is intercalated throughout the structure. And we can see that with the increasing particle number concentration of these particles, we see a commensurate increase in the palladium concentration. So we can use the palladium concentrations as a proxy for where the microplastics deposit. And so as a case study, what they did was they examined a wastewater treatment effluent. And so we can see the sludge layer here, and with depth, we can track where the palladium concentrations deposit, again, as a proxy for where the microplastics move through the environment. And so taking this idea, I wanted to develop a way that was similar in an approach, but I really wanted to get back that particle specific information. So can we track individual particle behavior in the environment? And so colleagues of mine in the chemistry department at Western Washington developed these particles where we have a latex core and we have gold nanoparticles intercalated throughout the structure of this core. And then that's capped with a polymethyl methacrylate shell. And so the important thing here is that the environment is seeing this plastic shell, but what we're tracking is going to be the gold signatures of the nanoparticles that are present in the core. And if we look at how big these particles end up being, they're at the 350 to 500 nanometer size range, which is well below where we're currently at with our state-of-the-art detection techniques. And so just some electron microscopy images to show you what these particles look like. Here we have some scanning tunneling electron microscope images. It's a little faint to see, but there's a lot of little nanoparticles embedded inside these particles here. When this is an example of a particle we wouldn't really use, but we can see a lot of the gold aggregated within the structure. And again, that's what we're going to be tracking with our approach. And so the approach we're using to track individual particles is a technique called Singleparticle ICPMS. And to explain that, conventional ICPMS has us taking an aquatic sample or an aqueous sample, and we usually dissolve it up. And so we have all these water droplets go into this plasma that generates ions, and those ions go through a mass analyzer and generate this constant signal. And that constant signal gets averaged, and we can use that as a way to quantify concentration of the element we're interested in. Now, single particle ICPMS, instead of dissolving up our sample, we instead let the particle run whole through the plasma. And so we have a bunch of empty droplets, and then occasionally we'll have a droplet that we're spraying into this plasma that contains our particle. When that particle goes into the plasma, we generate this big cloud of ions that moves through the instrument all at once. And so what we end up with is a bunch of blank signal corresponding to those empty droplets, and occasionally a big spike in intensity relating to our particle event. And so what we can do with this is we can convert that intensity of the particle event into a mass, and from that mass we can get a size. And so this is a particle sizing technique. And as in addition, all these spikes correspond to an individual particle detection event. And so it's a particle counting technique as well. And so we're going to use this technique to identify where our microplastics go in the environment. And so the benefit is that we get our spikes here, and we can convert that into a mass distribution. And so we can see here we get this relatively normal distribution of particle mass. And what we can use this for is we can maybe use this as a way to identify any changes in the aggregation behavior. So if two particles or microplastics come together, that's going to shift the mass distribution of the particles as a consequence. And so we can use this as a way to identify not only the number of particles, but also changes in the dispersity of the particle size distribution. And so some preliminary experiments that we've been working on, again, a monodispersed solution would have this nice normal distribution as shown here in just milliQ. If we expose our microplastics to different environmental media, such as T-clip or EPA mild hard water, we can see an increase in the mass distribution of the particles, potentially indicating aggregation. We can also further use this to better understand how microplastics partition into estuarine sediment. So an ongoing project we're working on right now is collecting sediment from Bellingham Bay. We are equilibrating our metal-doped microplastics with the sediment, letting it equilibrate overnight. And then what we're looking at is the amount of microplastics that are not bound to the sediment, those that come off easily after a brief sonication, so our loosely bound microplastics. And then we can fully dissolve our sediment to get back our mass balance of the microplastics that stayed in the sediment. So we can start to identify the behavior of microplastics when it comes to the partitioning of microplastics in our estuarine sediment. And so that was sort of a brief introduction into some of the work we're doing. And so moving forward, there's a couple other neat ideas coming about. One is that instead of using metal-doped microplastics, there might be the potential to instead look at a different isotope of carbon. So instead of looking for carbon-12, which would be pretty much impossible by ICPMS, we can maybe look at carbon-13 as a way to identify microplastic distribution. And so we can see here a paper from a colleague of mine, Melissa Velimirovich, identifying C13 as a possible way to identify the distribution of microplastics. One thing we need to do with these tracers, though, is we're still examining the physiochemical differences between these proxies and the native microplastics. And so by adding gold nanoparticles, how are we changing the density? Is it really going to behave like a native microplastic? Is it really that good of a tracer? We're still trying to really identify some of the pitfalls of this approach. But with that, there is a potential for maybe stable isotopic labeling. And so you can imagine if we have polymers of two different types, we could use the isotopes of a metal to label them and then use that isotopic labeling as a way to trace the behavior of the microplastics. And so with that, I would like to acknowledge students within my group, Rob Rauschendorfer, Star Summer, and Haley Sefi-Sear, as well as my collaborators, Professor David Ryder and Professor Steve Emery, and their students, as well as some seed grant funding from Western's Advanced Materials Science and Engineering Center. And with that, I would be happy to take any questions.
Great, thanks, Manuel. That was such an interesting presentation. I kind of feel like I might be several IQ points more intelligent than I was at the beginning of the presentation, I think. I know that there might have been a couple of questions come in, but I think, Heather, you might have a question that you wanted to ask to start with.
Sure, yes, thank you. That was awesome. I'm so glad you're doing this. Can you remind me and us, because I can't remember, how tiny a nanoparticle is relative to the diameter, say, of a hair? And then my question is, so what do you know from before your work about how these little particles, and I guess we can say like microplastics that are of this size, move around in the environment? Are they kind of like, if they're coming out of a wastewater treatment plant pipe and they're going out into, say, Bellingham Bay, are they zooming all around, or do they generally tend to settle out? What do you all know already?
Right, so to your first question, a human hair, I don't quite know the diameter of, I think that's 10 microns. And so a nanometer, in terms of nanometers, that would be 10,000 nanometers. And so a nanoparticle is 1 to 100 nanometers typically, but that's a very loose definition, and I could probably give a whole lecture on that. But suffice to say, nanomaterials and microplastics behave similarly. When it comes to the transport of microplastics, there's two things. You gave an example of wastewater treatment plants, which is a little tricky because wastewater treatment plants tend to sequester microplastics into the biosolids. So I imagine a lot of those microplastics are actually getting held up in the solid waste fraction of a treatment plant. However, if they are discharged into the aquatic environment, there's a couple of things that are going to determine its transport. Colloids, which is what microplastics are really governed by their settling velocity. And so in an aquatic system, that's going to be dependent on the density of the particle as well as the size. And so smaller particles and less dense particles stay suspended longer than more dense particles and larger particles. And so a PVC particle, polyvinyl chloride, which has a density of, I think, 1.3, is going to settle faster than a polystyrene particle, which has a density of 1.05, for instance. Other factors that are going to play a role is going to be things like salinity, because when we think about particles stay dispersed because they aren't attracting to each other. So particle attraction is a consequence of something that we call van der Waals forces. And those van der Waals forces are kept at bay by either electrostatic forces, so surface charge of the particles that are preventing them from aggregating, or steric forces, so physical barriers to them interacting. When they enter into a high saline environment, that surface charge that might be surrounding a particle gets reduced. It gets screened by all the other ions in the particle, which causes them to aggregate. So that's why we see big sort of we see particles settle out when they hit the estuary as a consequence. So there's a lot of things going on, I guess, when things get discharged out into the bay is what I'm trying to get across. But primarily what we should be thinking about is size, density, and the aquatic parameters that govern the colloidal behavior of these microplastics.
Okay, cool, thanks.
Manuel, we've had a question come in on the chat. Have you tried zonal centrifugation to study microplastic particles using their exceptional densities? We only have 90 seconds left, and I wonder for people like me on the call, if you can just translate that into simpler English, that would be great.
I'll be honest, I haven't heard of a term of zonal centrifugation. I will say that one thing that we are doing is we are trying to examine the use of things like centrifugation, settling as a way to separate particles from sediment, from aquatic systems. This is one of those things that we're actually trying to attempt now to look at how the tracers behave relative to the native microplastics. Do the densities match up, or do they not match up, and what does that mean? And so there's a lot of potential here to use things like centrifugation. And one technique I'm also trying to develop right now is some density gradient centrifugation techniques as a way to also better separate microplastics from the surrounding media so that we can get a better handle on their characterization.